anticipating some other people will still join, but I want to try to keep to time. Lauren, you're online, right?
Yes.
Okay, great, thanks. So yeah, this is our monthly CASP special interest group on ensembles of conformations, trying to explore how CASP might be able to impact the general field of protein dynamics and conformational switching. This is the summary of what we'll be doing today in the hour. I'll give a little bit of an introduction, which is similar to ones I've done before, just to give a sense of what the CASP special interest group, this CASP special interest group is about. And then we're very fortunate to have Lauren Porter talk today from the National Institutes of Health, National Library of Medicine. And she'll be talking about evolutionary selection of proteins with two folds. And then we'll have 15 or maybe 20 minutes for just open discussion and questions about Lauren's talks and ideas, and also about the general challenge of how we can implement a CASP experiment in this domain. Comments in the chat are welcome, and they'll be recorded. Actually, we'll make a video of the presentation, which will become available, publicly available. And the PowerPoints are available if you join the mailing list. We make the PowerPoints available to people that way. And there's also a LinkedIn group where we post relevant papers as they come up in this topic area. So this is our kind of our vision statement, recognizing that biomolecules don't have static structures, but rather adopt a distribution of states that underlie their functions. The CASP SIG aims to evaluate if we can do a critical kind of blind assessment of the predictions or modeling of conformational ensembles or multiple conformational states, whether such an activity is even feasible. It's not the same as having a crystal structure as the gold standard, and we're exploring how to do this. The SIG will consider developments in the field of structural and computational biology that are anticipated in the coming years and define protocols for assessing the accuracy of multiple or ensemble model predictions. And so conformational ensembles simply means the concept of multiple conformational states. It might be as simple as a two-state flipping, the classic case being adenylate kinase, to wide distributions of conformations we call intrinsically disordered proteins or interdomain protein linkers. There's actually been some discussion on this point, whether it's fair to use the word ensembles if you're not talking about the statistical, mechanical, canonical ensembles or constant P&T type ensembles, or you're talking about Boltzmann weighted distributions. All of those in the context here are kinds of ensembles, but ensembles is used in the general term. Maybe we don't know the populations, we just know the two states. That's still an ensemble in the way we think about it. But of course, to know populations is very important too, and that's one of the challenges. So this is our organizing committee. This was put together by the CASP organizers, John Molt, Christoph Fidelis, and Andrei Christofovich. But we have people who have been involved with CASP over the years and or have a strong interest in this area that are listed here. And if there are other people who should be part of this in the future, you can send an email to this. This actual website is particularly being used for this CASP SIG. In the CASP 15, we did have a couple of targets as multiple conformational states, some from cryo-EM and some from NMR. There are other methods that could be used. And in this paper, we explored how do we do this? How do we define the targets? How do we define the CASP experiment? How do we assess the targets? It says in press, it's actually out in the special issue of December of CASP 15, which has all the papers. It's also open access for people to look at. So if people are interested in maybe participating in this as predictors or providing targets, this is a good starting point to get a sense of what the experiment is about. And we're going to do this for CASP 16. Probably targets will be available on the time frame of May. And the CASP season runs from about May to August. I'm not sure the exact time this year. I don't know how many we'll have. We're seeking such blind targets where we have experimental data, and we're going to focus on validating not against the, say, experimentalists' models, which have to interpret the data, but rather to validate against the data itself, and that's a bit challenging. So we have to collect the data to be able to validate against and ask how models, and this is a challenge. We may run into issues. So the kind of cases we're looking for are multiple conformational states of a single protein, like adenylate kinase cases that are not yet in the public domain, fold flipping, ligand-induced conformational changes, allosteric shifts that occur upon complex formation, environmental-induced shifts due to pH or temperature or salt. Interdomain linkers are a kind of intrinsically disordered region, which are very important. And I think we have some data sets for that we'll have for this CASP. And maybe probably at least a few more traditional kind of IDP proteins. So that's what we hope we'll do for CASP 16 in the spring. And very much this SIG in the spring will be focused on those methods, those kinds of data, as well as innovations like Laura's going to talk about today, new methods. So the next meeting is January 3rd, but we haven't identified the speaker yet. It's the first Wednesday of each month at this time. And there's also a parallel AI special interest group of CASP, which is really focused specifically on AI methods, both of modeling and of de novo protein design. The next meeting of that is on December 13th, which is a general discussion. And then on this January 10th, Philip Kim will talk about machine learning methods for de novo protein and peptide design. It's a very exciting area. Arnie Ellefson organizes this, and here's the homepage he's set up for it. And then there's also a CASP SIG on modeling, predicting of RNA structures that is organized by Rachel Kresh. She has the schedule published here, and you can join their mailing list. And the exact date actually changes. You really have to check the mailing list to be sure about the right date. These are upcoming speakers. Yeah, that's kind of my introduction. And now we're going to go into the presentation. So Lauren, do you want to take over the screen, and then I'll do a short introduction as you put up your first slide?
Sure.
Okay. So
all right.
Thanks, Laura, for joining us. And so Laura is Stadtman Investigator in the Office of Director at the National Library of Medicine in the National Institutes of Health. And she also has a joint appointment in the National Heart, Lung, Blood Institute. And Dr. Porter has had a significant history, about 10 years or so, in studying fold switching proteins, both computationally and experimentally. She did PhD work with George Rose at Johns Hopkins University, and then a postdoc with Phil Bryan and John Orban, University of Maryland. And then I guess as a second postdoc or more research with Lauren Luger at HHMI in Maryland. She's been recipient of the Maryland Academy of Sciences Outstanding Young Investigator Award and a National Research Service Award. And she's actually making a significant impact in this field. So thanks, Lauren. Do you want to just go ahead? And I think I said Laura, but it's Lauren. I'm sorry.
That's okay. No problem. Thank you, Guy. And thank you all for the invitation to speak. I am really excited to be talking to this audience at this time because we've been doing a lot of benchmarking that we are about to put out. And I would love to get your feedback and have discussion about what we see and what your thoughts are. So Guy, it sounds like there's plenty of time for discussion. So I'd love to hear your input. But first, let's talk about a little bit of background. So As you know, if you look at the RefSeq database right now, there are about 290 million curated non-redundant protein sequences. And so this is a subset of the protein universe. It's kind of a higher quality subset. And within that subset, I should also say, so that we know from this large data set that the structure, the sequence of a protein is important for its structure. And this is true for both folded proteins, such as this NUSG binding domain for the ribosome, and also for intrinsically disordered proteins, such as this human SOX5 domain, which is involved in signaling. Now, if we look at the protein universe and look at what we know experimentally, we can see that there are about 70,000 non-redundant proteins with solved structures, 5 angstroms or less, solved by any method. And so the question is, how well do these 70,000 experimentally determined structures represent the protein universe? And what I would say is, we certainly have gotten a good representation, but I also think that we have some more to learn. And let me show you an example of why. So as Guy mentioned, the kind of proteins that I work on, my interest was piqued by Phil Bryan and John Orban's work in the early 2000s is on these fold switching proteins. And specifically, I'm interested in naturally occurring proteins that change, remodel their secondary and tertiary structures in response to cellular stimuli. So here's an example of one. This is E. coli RFAH. It has a conserved N-terminal domain that you can see in gray, and you can see that its C-terminal domain in the auto-inhibited state is helical, but upon binding both RNA polymerase and a specific DNA sequence called OPS, this protein dramatically remodels its secondary structure and switches its fold to a beta sheet, which binds the ribosome, fostering efficient translation. So this is one amino acid sequence that has 2 folds that are important for 2 different functions. And when I was in Lauren Luger's lab, we actually benchmarked the PDB and found approximately 100 examples of proteins with identical or nearly identical sequences that change their fold and functions in response to cellular stimuli. And it turns out that these fold switching proteins, I mean, I'm interested in them just because I've always been amazed with protein folding, but it turns out that these fold switching proteins are involved in a number of human diseases. So here are a few examples: tuberculosis, cancer, Alzheimer's, gastroenteritis, and even a COVID-19 protein has recently been found to switch folds. Not only that, but just this year, it was found that minor sequence modifications can actually cause major changes in protein structure. So this is a human protein called BCCIP. It has 2 isoforms, the beta isoform, which is conserved all the way from yeast to humans, and its structure was deposited in the PDB quite a few years ago now. And below here is a diagram of its 2 sequences. So the dark parts are conserved, so they're 100% identical all the way out to here. But then just with 1 exon substitution, comprising the last about 20% of the sequence, causes the protein to completely change its fold in the beta and alpha isoform. What I mean by that is if you were to superimpose these 2 structures and do an all-atom RMSD, it would be 13 angstroms. And the group who solved this alpha isoform, which has only been found in primates and a few other species, solved it 3 different ways, twice by cryo-EM and once by crystallography, got the same answer all 3 times. And when they searched this fold using DALI in the PDB, the closest thing they could find was 10 angstroms away. So this really is a new fold, it seems, that is caused by a very relatively minor sequence modification that one would expect would not have such a large change on the structures, on the protein structure and on its function. BCCIP alpha has binding activities that BCC beta doesn't have, and these are related to cancer. So the question that my research wants to address is, how do minor changes in sequence or environment cause proteins to switch their folds and functions? And I think the significance of this is, hopefully, by studying this in more detail, we'll come out with a more complete understanding of protein folding and dynamics. Maybe we'll be able to discover new biological pathways. Fold switching may explain some biological processes that we don't understand before. It's certainly been shown to regulate, for example, the circadian clock of cyanobacteria. It does have important biological functions. It may explain more. Mutations can lead to alternative folded states. We know this from Phil Bryan and John Orban's work, but also in natural proteins. I just showed you an example where alternative splicing switches a protein fold. There's also an example of one SNP in another human cancer protein, a D83V mutation that switches a protein from an alpha helix to a beta sheet, presumably inactivating it. And that's the most common cancer-associated mutation in that particular protein. And it may also be that post-translational modifications could cause proteins to switch their folds and functions. But the issue is that there are barriers to progress. As I mentioned before, so far, we were able to find approximately 100 examples over the entire PDB of proteins that switch their folds, meaning like they remodel their secondary and tertiary structure in response to a cellular stimuli, not something like crystal contacts. Second of all, fold switching is difficult to predict. And finally, fold switching proteins can be very difficult to handle experimentally. This is a major challenge in my lab working with RFAH. Here's an example. You can see 3 different NMR tubes of this protein, and at the bottom you see snow. And this happens a lot. The proteins that we work on crash out of solution often and easily. Some of them are only soluble up to 2 micromolar. That makes it very difficult to do much structural characterization at all. But given the audience, I know you're probably most interested in talking about predictions. And so what I want to say is that fold switching is difficult to predict. So my group published this paper about 1.5 years ago. So it's a bit outdated now with all of the work that's been done on AlphaFold since it was published. But back when we published it, as I said, fold switching proteins have 2 different conformations. And so we used AlphaFold to predict, to see if it could predict both conformations, just using very simple techniques, just using like the standard technique, and then also testing with shallow MSAs. And you can see if we look at the prediction accuracy of one fold versus the other, AlphaFold was at this time very highly biased to predict the dominant fold and miss the alternative fold. Out of the 93 proteins that we tested, we were only able to get AlphaFold to capture 8. Now, as I said before, a lot of new stuff has come out with AlphaFold. People are interested in predicting alternative conformations. And so we thought it was really important to address this question in more detail with newer versions of AlphaFold and newer sampling methods. And so here's some of the new benchmarking data that we are getting ready to submit. So here's our initial data with AlphaFold 2.2.0. You can see 2.3.1 does better than 2.2.0 by a little bit. The multimer model gets us 1 extra prediction out of the 93. We ran Speech AF by the Makarov lab, where you knock out co-evolutionary signal in a multiple sequence alignment. We generated 76,000 different structures of our 93 fold switching proteins. It captured 7 out of the 93. We also used AF cluster and actually sampled quite a bit more than they did, with more MSAs and generating more structures. In total, we generated almost 210,000 different structures using 2 different versions of AlphaFold, 2.2.0 and 2.3.1. And you can see that it captured some, but it was a modest success. And if you put everything together over almost 300,000 structures, we capture just under a quarter of the fold switching proteins that we have benchmarked. So this is better than what we saw with AF 2.2.0, but still, I would say we have a long way to go in being able to predict fold switching proteins. Now, if you might ask, well, it might help to talk some about how these methods work. And here in particular, I want to focus on AF clusters since it was the most successful in predicting and also has just been published. So as you may know, the way AF cluster works is an MSA is generated using collab fold, and then it's clustered into small clusters by sequence similarity. The authors made sequence clusters that generally range from about 4 to 10 different sequences. And instead of putting in the deep MSA, they put in these small MSAs to see if they could predict alternative conformations. And they found that they did in several cases, mostly with the circadian clock protein CASP. Now, we, having done all this benchmarking, we tested this without using the sequence clusters at all. We just inputted single sequences, and we found that we were able to reproduce their results without sequence clusters. And so from this, we believe that AlphaFold is actually learned these structures and is associating sequences similar to the structures that it's learned with the structures in its training set. And we've tried this in multiple ways with collab fold, with AlphaFold, different versions of AlphaFold, and we are able to reproduce these results from single sequences. And sometimes just using a 1 sequence MSA does better than the 10 sequence MSA clusters. So again, this suggests to us that AlphaFold is associating structures it already knows with similar sequences. And we actually found this about 6 months ago in a related work on alternative conformations, where we found a natural evolutionary pathway between 2 protein folds. And unfortunately, I don't have time to get into the data here, but if you want to discuss, we can at the end. But what we did is we found that these 2 different response regulator proteins, where one has a C-terminal alpha helix and one has a C-terminal beta sheet, likely had a common ancestor. And when we did ancestral sequence reconstruction and ran it through AlphaFold, if you look at these red C-terminal secondary structure elements, AlphaFold is also predicting switches from alpha helix to beta sheet. So again, there are a lot of response regulators in its training set. And so it seems, again, to be associating structures it already knows with sequences that are similar. To look at this in more detail, we can look at a specific example from AF cluster. So this cluster was taken directly from their GitHub site. If you put it through MSA Transformer, you end up getting this contact map. And the contacts that are really important, as you can see, are circled in red, orange, and purple. Those correspond to the unique parts of the fold that they looked at, the CASP fold. And when you input this sequence, again, from their GitHub page, directly into AlphaFold using the same methods reported in their manuscript, you end up with this high confidence prediction. Now, the problem is MSA Transformer uses 2 things to make these contact maps. It uses both sequence similarity and coevolution, but it's possible to be able to tell which one it's using by 2 tests. So the way the MSA Transformer was trained, if you reorder the sequences in the MSA, that is supposed to help get rid of the sequence similarity component, because MSA Transformer was trained on curated MSAs where the evolutionary distance was preserved. It wasn't scrambled. And so the authors say of MSA Transformers say in their paper that scrambling the order of the sequences should be able to preserve the covariance or the coevolutionary signal, but it should get rid of sequence similarity. By contrast, if you scramble each individual column randomly, you should be able to knock out the covariance signal and just be looking at similarity. And so when you do this test, what you can see is that the unique contacts corresponding to this fold all seem to arise from sequence similarity, not from coevolution. And you might say, well, MSA transformer and AlphaFold are not exactly the same thing. Maybe AlphaFold is seeing something different. And so to test this, we put this scrambled MSA, the same thing that we used to generate this contact map, into AlphaFold. And we ended up getting a slightly higher confidence prediction of the same sequence. So putting this all together, what this suggests to us is that AlphaFold is doing a really nice job of kind of homology modeling, relating sequences in structures that it's seen to other sequences. But the concern here is it may not do super well at predicting things that are new. And we actually tested this because there are two new fold switching proteins that we know of that were published after AlphaFold was trained. Both of they came out in 2023. And so we ran AF cluster on both of these new fold switching proteins. And what you can see is even though we generate thousands of structures for these sequences using many different sequence clusters, generating 50 sequences per cluster using many random seeds, it only captures one of the two folds. We never see it capture the alternative, even though we did a lot of sampling. And so this is a little bit discouraging to us about how much AlphaFold may be able to do predicting new fold switchers that it hasn't already seen in its database. So it still may be able to predict new fold switching events like we've shown, and as Waymont Steele and colleagues propose with their DSBE sequence, it may be able to predict fold switching in related sequences that AlphaFold has already seen. But personally, I'm not optimistic about its ability to predict something new. And one other concern that I think I need to voice is that the way AF cluster works is that it breaks down these sequences into small clusters and then basically uses those clusters almost as it uses those clusters with a given sequence to be able to predict a fold. But then the question is, well, how do you know if it predicts one fold or the other? How are you going to know whether the prediction is correct? And unfortunately, there are times when AF cluster confidently predicts incorrect folds. So here's the structure that's up on their GitHub site of Chi-B from S. elongatus. If you look at it, you can see the confidence is not very high. The average PLDDT is 55. We took their Chi-B TV4, which was predicted in a different fold with high confidence in a clade that should not switch folds. And we superimposed it on a cluster that does bias towards the fold switched conformation, which is incorrect for this sequence. And you can see that AF cluster is predicting the incorrect fold of this sequence with higher confidence than it predicts the correct fold of another actual fold switcher that has lower confidence. So we're concerned that using this as a general method may give some misleading results. And this is actually consistent with what we see in our benchmarking over all protein fold switchers overall. So this is true for AlphaFold 2 and also AF cluster. If we look at its confidence metrics, and here I'm showing you PLDDT scores, if we look at all predictions, regardless of PLDDT. You can see, so top 1 would be best PLDDT among all, top 10 is top 10 among all, and then all is all. You can see that we get some fold 1 and some fold 2. We do okay if we don't pay attention to PLDDT scores. But when we start paying attention to PLDDT scores, we see less and less of especially the alternative conformation. So medium is, I think, 70% of all amino acids with PLDDTs of 70 or higher. Good is 80% with 70 or higher. High is 90% with 70 or higher. And you can see that as the PLDDT scores go up, more experimentally unobserved conformations are observed, and fewer of fold 1 and fold 2 are observed. And this is especially concerning for fold 2 because we're talking about alternative conformations. So here's just another way to visualize the data. If we look at all, looking at all for fold 2, you can see this is where we get the most frequent predictions of fold 2. But as the PLDDT metric, as we use higher and higher PLDDT metrics, we see fewer and fewer predictions of the alternative folds. Whereas we end up seeing more and more predictions of other folds that have not been observed experimentally. So with all of these observations, we wanted to take a different approach and just get a sense, like, how predictable is fold switching anyway? Maybe this is just, I mean, obviously this is a really hard problem. And so why might AlphaFold be missing fold switching? I mean, even with 300,000 structures, it captures less than 25% of fold switching proteins. So why? Well, for one thing, it could just be that these missed conformations are a product of mutational drift. They don't co-evolve. And this is actually what Danny Tawfik had suggested not that long ago, back in the like the 2010s, that these fold switched conformations are not selected. They're sort of just mutational byproducts that go away. The second option is that coevolutionary signals of missed conformations may be there, but they could be too subtle for detection. And finally, there could be some other unknown reason. This is the hard thing about using AI. It could have to do with how AlphaFold was trained. But our working hypothesis was that the coevolutionary signals of misconformations could be too subtle for detection. So back in 2022, we used JPRED, so another sequence similarity-based tool, to predict correctly fold switching in 10 out of 10 NUSG-like proteins that were 30% identical or less. So that's a pretty hard metric to hit. By comparison, the AF cluster predictions were all 47% or higher identical to PDB targets. And what we noticed when we did this study is that the way that we were able to infer fold switching was by using JPRED, first looking at the full sequence, where we always got beta sheet prediction in its fold switching C-terminal domain, which goes from beta sheet to alpha helix. But if we only put in MSAs from the C-terminal domain, we were getting subfamily specific signals that all of a sudden were showing alpha helical predictions. And so what we would do is compare the JPRED prediction for full length with the cropped and see, all right, if there's enough alpha helix, we infer fold switching. And that worked with experiments 10 out of 10 times for highly diverse NUSG sequences. And what we noticed is that these full-length sequences, their depth and diversity measured by NEF were substantially more than these cropped sequences, which made sense to us given how fold switching has been found to evolve. So what's usually happens is you have this superfamily, which would correspond to the full-length deep NEF sequences, where some proteins switch folds and other ones don't, but all of the proteins assume one conformation. In this case, all NUSG proteins, whether they switch folds or not, will assume the beta sheet conformation. But it would only be in the specific clades that switch folds where you would see this alpha helical prediction. And so our approach was to take our original MSA, which is the deep one, and filter the sequences by similarity until all we're left with is a subfamily MSA more specific to our target of interest. And this is important because it keeps our sequence near the sequences that it evolved to in evolutionary time. The problem is if you start comparing it to sequences further away, that's when incorrect predictions can happen. And so we wanted to keep everything close in their clades. And so we looked at RFAH, as I mentioned, here's its N-terminal domain in gray, and here's its fold switching C-terminal domain that you can see in black. And as I mentioned before, upon binding both RNA polymerase and a specific DNA sequence called OPS, it switches to this white C-terminal domain, and you'll see why I did this coloring. So it's a complete alpha helix to beta sheet switch. Now, back when we did our work, the Nature Communications paper I referenced before, we found a protein that we assigned by NMR called variant 5, whose experimentally consistent C-terminal domain was helical. But as you can see, alpha fold 2, Rosetta fold, ESM2, and RGN2 all predict this ground state beta sheet fold. They don't pick up on the correct conformation. Now, let's go back to our MSA. If we run Gremlin, then we can get coevolutionary signals. But let me explain my contact maps first, because you can see that they're asymmetric. On the upper diagonal is the contact map that corresponds to the beta sheet conformation only. The light gray contacts are experimentally determined contacts that are unique to the beta sheet fold. And the medium gray are conserved between the two structures. On the lower diagonal, again, we've got the conserved medium gray, but then we also have black, which are unique to this alpha helical fold. So if we run Gremlin on our deep MSA, so all the sequences together, you can see that it gives us coevolutionary signal that corresponds pretty well to this beta sheet, but we hardly get any signal for the alpha helix. Digging deeper into subfamilies, you start seeing predictions, which I should say, the predictions are teal. So you start seeing predictions that correspond to both alpha helix and beta sheet that weren't seen before. You also see noise, which I'll talk about in a minute. So, but why stop with just like the deepest MSA and the shallowest subfamily specific MSA that we can get within the noise of what Gremlin can do? And why just use Gremlin? Why can't we combine other methods for predicting covariance or coevolution? So when we do this, we end up seeing good signal enhancement for this variant 5. So you can see that when we stack up a bunch of coevolutionary predictions from many MSAs of many different depths, you can see a lot of contacts corresponding to both conformations. And but the problem is you can see all this noise. Now the noise is sparsely distributed, so we can actually eliminate a lot of it using density-based scanning, and we still preserve some coevolutionary signal for both folds. And the p-value, the probability of being able to get this good of a fit to the two folds is very low, suggesting that coevolution is a very good explanation for getting both of these folds. And in other words, what that means is that evolution has actually selected to preserve both folds in this variant 5. So that's nice for variant 5, but how about fold switching proteins in general? So we tested this on all of our fold switching proteins, but we couldn't get deep enough multiple sequence alignments for all of them. We could only get deep enough ones for 56. But you can see that in these 56 cases, if you compare for the alternative fold, our full pipeline with the original analysis, which would be predictions from MSA transformer and Gremlin together using deep MSAs, we get on average about 200% signal enhancement for the alternative fold. suggesting that generally speaking, evolution has selected for both folds of fold switching proteins. And this is unique to fold switching proteins. It does not apply to single folding proteins as well. So this has answered a question that I've wondered about for a long time. People have asked, like, aren't fold switching proteins basically just intrinsically disordered proteins, just kind of another thing? And I think this pretty clearly says that the answer is no, because IDPs are pretty much not observed to co-evolve, or if they do, they co-evolve with exogenous factors like their binding partners. That's something that Peter Tompa found in a paper. It's the only paper I've been able to find on co-evolution of IDPs is them and their cofactors together. But there's only one co-evolutionary signal that seems to be exogenously determined by cofactors. And whereas with ubiquitin, with like a single folder, there's only one co-evolutionary signal unique to the one fold. But with RFAH and with other fold switching proteins, we actually see co-evolutionary signals for two folds. So this does seem to distinguish fold switchers from the other sorts of proteins. Now, can we apply this and be able to predict three-dimensional structures? So if you look at this variant 5, where I showed you before, if you put in the deep multiple sequence alignment, to AlphaFold, it predicts only the beta sheet conformation. I looked at this and I asked, okay, how many unique contacts do we get from our coevolution that correspond uniquely just to the beta sheet fold? So we're only looking at unique beta sheet contacts. And you can see there are like 21 of them. But if we look at, and then if we ask, okay, well, how many contacts are unique to the alpha helical fold? There are only 4. So this is consistent with our hypothesis that the coevolutionary signal may really be there. It's just too subtle for AlphaFold to detect. So then we can use the speech AF method that was suggested, where we knock out the coevolutionary signal unique to the beta roll fold. And I should say, we don't have to use alanines. We could just repeat the input sequence again and again and again. So the result that I'm about to show you is not because of alanines, but when we knock out coevolution, we actually do end up predicting the alternative fold. Now, it may not be because of coevolution based on all the other data that I've shown you. I mean, we made these predictions using our coevolutionary contacts. AlphaFold may be using something else to predict them, but using our coevolutionary signal, we were able to predict the alternative conformation with an experimentally, that's consistent with experiments. So these are alpha helix and beta sheet estimates based on NMR assignments that we did. And you can see that the regions that correspond to helix in the prediction match what we see from our NMR. And again, I'll mention this protein is 29% identical to anything in the PDB. So it's a pretty hard prediction to make. So as I said before, I hope you believe me that fold switching it's a difficult problem. It's difficult to predict. And AlphaFold at this point, at its best, as far as we can see, can predict maybe about a quarter of the fold switching proteins that we've benchmarked. ACE takes us to more like 60%, but we still have a long way to go. I think there's a lot about fold switching that we have yet to learn and have yet to understand. But we do feel optimistic that we can use ACE with some confidence to be able to predict new fold switching proteins. And Joseph from my lab has actually run predictions on the entire E. coli proteome. And we're trying to be very careful before we put this data out, before we put out these predictions. But at this point, we're estimating that approximately 5% of the E. coli proteome may switch folds. And so again, fold switching isn't just a kind of an evolutionary byproduct that's unimportant. It may be a lot more common than we expect. I mean, clearly not the norm, but maybe important. So with that, I'd like to thank all the members of my lab for their hard work on these projects, especially Devlina, Joseph, and Ethan, who have done the work that you've seen here. And I'm happy to take your questions.
Well, thank you. That was really great. I want to just open the floor for people to ask questions, and if you want to put them in the chat, I can call on you, or you can just speak up.
Yeah. Hi, Laura. Can I start?
Yeah,
Sure.
and it's probably good. I just thought of it. If people just identify themselves when they start talking.
Yes, I will. So I'm Frank, and I work at the Free University of Brussels. I'm also part of this this CASP group. Really nice talk, Lauren. So I was wondering, do you think that experimentally speaking in the folds, like, so, you know, if you have fold 1, fold 2, if you look at fold 1, is there anything that you can see, like with NMR, for example, or in the dynamics that might indicate that there is, you know, that it might be able to change fold or not? That has anyone ever looked at that, or is there?
So are you asking like, is there some way that we could potentially screen for fold switching? Is that what you really want to know, or about NMR?
Well, yeah, but also just like, I mean, is there something, because I mean, you know, if it can switch secondary structure, it probably means that one of those is maybe not as stable as a, you know, a full-on alpha helix.
That is true. So I will tell you, so our variant 5, where I showed you the assignments, was a beast to handle. Actually, at first, I only double labeled it. So it's only 168 amino acids. And I double labeled it at first. And the T2 relaxations were so fast, we couldn't get good enough 2D data to be able to make the assignment. So I had to go back and deuterate it. And that allowed us to be able to make those assignments, which you can still see like aren't awesome, but it's like that is what it took for just 168 residue protein. And so I do think these proteins resist structure characterization, which may be why they could be underrepresented in the PDB. To address the larger question, I do think experimental methods to screen for this are critical. And so we're working with 2 different groups to develop 2 different methods. Unfortunately, at this time, NMR is a little difficult because you can't really do high throughput experiments with NMR, and we want something that we can do with more, be able to test more. And so we have 2 different methods. Leslie Ronish in my lab is developing a FRET-based method that we have very promising results for with RFAH. So this would work in cases where we have 2 structure predictions where the end-to-end distances of the predicted conformations are very different from one another, which is the way RFAH and NUSG work. So that's one case. The other case, Joey Thole in my lab is working in collaboration with Susan Marques's group to use a hydrogen deuterium exchange mass spec based method. And that's in progress. It looks like, of course, we're testing everything on RFAH and NUSG first because it's a system that we know. So that is looking good so far. But what would be really great with the HGX MS is to be able to test it on things where they may not have different end-to-end distances and you can't tell anything with CD. So for instance, I think Chi-B would be a great candidate in that case because it has both conformations have similar alpha and beta content. So you can't use CD for that like we did with RFAH. And also their end-to-end distances aren't that different, so you couldn't use FRET. So we're hoping that by using a couple of different methods in different cases, we may be able to fish more fold switchers out of genomes. But it's going to take a little time to develop those methods.
Yeah, I can imagine. Thanks. Yeah. And if I may continue on that, so and in the structure itself, do you think there's something there? Because, like, you know, you have, for example, that frustration method from Valencia's lab.
Are you talking about the frustratometer?
Yeah. Is there anything there, or it doesn't really show?
No, there's nothing there.
Okay. Yeah. So it really is something that is sort of emergent in the sequence that is not obvious from the like the single atomic model that you get out of
Yeah, unfortunately not. I think this is just a hard problem on every level. It's hard computationally, it's hard experimentally, and we're really just learning, trying to figure out as much as we can right now.
Yeah. Okay. Thanks. I might actually send you something because we looked at some of these proteins a while back using the predictors that we have, which are sequence based and sort of bit more, what the protein can do. And we did see interesting signals in the regions that switch fold. So maybe there's something you could do with that.
Yeah, thank you.
All right. Thanks.
We have some raised hands, and we'll use this
Yeah.
method. Shoshona, why don't you go next?
So I was wondering about the influence of the contexts of these different folds. In other words, if obviously the context stabilizes one of the folds versus another.
Yes.
So how is that taken into account, and can that be taken into account?
Well, that's a great question. So I couldn't because of time, I couldn't get into that in my talk in general, but you may have noticed that I said something about using AF2 multimer to be able to make predictions of alternative conformations. And so we actually use the multimer model on single sequences without binding partners and with binding partners.
Okay.
And what we found is the binding partners provide no extra information to AlphaFold.
Right, because AlphaFold doesn't consider that. Does it consider the binding partner in the prediction?
Yes, it does. You
So
can.
you're basically trying to predict the complex.
Yes,
between the two.
exactly. So if, for instance, you have like RFAH and RFAH in complex with the transcription elongation complex, you can give it, both RFAH on its own and RFAH with transcription elongation complex. So that's what we did with the multimer model. Tried to see if the multimer model, if you gave information about binding partners, could it predict the alternative conformation? And we found that for AlphaFold, giving that extra information about binding partners did not enhance predictions at all.
So what you are saying, just one last thing, reacting to your comment, what you are saying is that it doesn't predict the complex correctly.
Sometimes it does, but not usually.
Yeah, that's what I'm. Yeah.
But when it does, it can predict the correct conformation without information about binding partner.
Okay, thanks.
Yes.
I think actually Ray Blind was next, and then Keith. So go ahead, Ray.
Sure. Ray Blind I'm at Vanderbilt. I'm curious if in the MSA, rather than replacing it with just alanines, if you could shift between these two states, the two folded states, by stabilizing, by putting in the MSA like a disulfide bridge or a salt bridge or something like that.
Possibly, though, that would be a different protein.
Yeah.
Well, yeah.
Keith?
Yeah, hi, that was a great talk. I'm a postdoc here at RPI. And my question is again also related to the editing of the MSA.
Yes.
So you had this example where you masked out columns for one of the states to bias the AlphaFold to get to the other state.
Yes.
But in the ACE paper,
Yes.
there was this comment or note that this masking, that this editing of the MSA doesn't always work.
It does not.
And so I wanted to get your thoughts as to why that could be the case.
Yeah. So no, that's a great question. So this is going back to what I was trying to say before. So in my mind, the masking of the MSA was just a demonstration that using our coevolutionary contacts did have some predictive power, but it is not a magic bullet in any sense. And I think in part, it's because, as I said before, as far as I can tell, AlphaFold seems to be inferring alternative protein conformations from sequence similarity, not from coevolution. And so it has to have seen the alternative conformation in its training. And the masking has to help it see the alternative conformation in its training. So, the masking the MSA is not is not a magic bullet. And that's actually why we didn't, in our paper, we didn't propose it as a general method. If you notice, instead, what we proposed is using AlphaFold with full MSA to get one structure, and then ESM fold to get another, and then seeing how our contact maps matched up. We believe that that may be a more reliable method, because once you start messing with the MSA in any number of different ways, like AlphaFold wasn't really made for that. And so then it's like you're messing with it and you're not really sure what you're going to get out. So we feel more confident about using AlphaFold with its full MSAs, the way that it was built to work. And that is how we predicted alternative conformations of that response regulator family that I showed you earlier with the fold switching. We did nothing to the MSA at all.
Excellent, thanks. Yeah, that was it. Thank you.
Thank you.
So Shoshona, and then actually I have a question after Shoshona. Go ahead.
Okay, that's a kind of a naive question. Are all your fold switching proteins monomers?
No, many of them are multimers, either homo-oligomers or two switch folds. They have to bind to another protein to form a hetero.
So do you take that into account in your approach?
In my approach for
I don't know, whatever, if you know that, are you to predict, for example, the influence of another binder, and you have actually a dimer, you should use the three proteins together.
So that does get difficult, because as you
I know.
Yeah, so but I would say, so like for RFAH, for instance, we don't need information about the binder to see dual fold coevolution. The single sequence is enough. And that's better for us, because as you know, you dilute signal as you need more and more sequences.
Right, exactly. Yeah.
Alignment. And so we're encouraged that you can, without any information about binder, often, not always, see alternative conformations. You can, and we're actually right now building a neural network that will help us potentially distinguish homo-oligomeric contacts, which we also see from fold switching contacts, because that is a very challenging. Yeah. So we recognize, like, this is fraught, and we're doing everything we can to try to rule out other possibilities.
Interesting. Thank you.
Yeah.
So Lauren, you pointed out that, I guess it's together with Lauren Luger, you developed this a database of fold switching pairs to validate against. And then you pointed out that with various methods, you only get about 20 or 30% coverage. And then you also pointed out that some cases, in many cases, you actually predict other folds that are not amongst the alternative folds.
Correct.
And so we view those as somehow, I don't know, false positives. But is it possible that those other folds are real? They just haven't been seen yet experimentally.
So I can only speak to that with experimental evidence from an N of 1, okay? But I'll tell you what I know. A group from Germany did CEST experiments on RFAH, and they were able to observe a sparsely populated intermediate of the alpha helical, from beta sheet going to alpha helix. They were able to characterize it, and the alternative conformations that AlphaFold predicts do not match that intermediate. So that's an N of 1. That's all I have.
Okay. Well, I think it could be in some cases, and it's a question of figuring out what's real or not. It's actually giving us insights into other structures that just haven't yet been achieved experimentally. And then I have another question. So ESM fold is using a single sequence and doing a large language model prediction. So
Yes.
What's your experience with generating multiple conformational states with ESM fold?
Yeah, we've been working on that some. So I should tell you, like, I'm not sure ESM fold. So Sergey Ovchinnikov has pointed out that the structure module for ESM fold is highly overtrained. And we see that. So for instance, if you put the sequence of RFAH just through the language model, so the ESM2 language model, and you plot the contact map before it goes into the structure module, it's all beta sheet. There's no alpha helix at all. Then if you watch after it goes through the structure module, step by step, as it comes out, you see more and more helix coming out. So that can't be from the contacts. It has to be from something that structure module hasn't learned. And so, again, I think we're going to run into the same problem with ESM1B or with ESM2, where we're likely to maybe be able to use it to predict alternative conformations in the training set. And we can do that. We have successfully done that with this variant and other proteins by doing some sequence masking, which ESM makes very easy. And something that I like about ESM is it's very easy to follow. Like, it's easy to get into the code and see what's going on under the hood. So I like that. But I think we are likely to run into the same problem with ESM, that we're going to be limited to what's in the training set. It can't predict BCCIP alpha when we mask, for instance.
Well, I do want to end on time, but since I took the last question, I will allow if someone else wants to ask one more question before we wrap up. There's one more. And if there's not, thank you, Lauren. Thank you very much for taking the time and effort to do it. It was very relevant to our discussion. And I hope that this community can talk to their friends and their colleagues and help us to identify appropriate targets for our CASP 16 blind experiment. So if you have any ideas, reach out to us and we'll figure how we can make that work. Okay, so somebody asked about translational modifications. Do you want to just answer that quickly, Lauren? Just take a minute.
So I am, like, my gut is confident that sometimes protein post-translational modifications do flip protein folds. Julie Foreman K, the closest story I know of is Julie Foreman K, showing that that they do switch intrinsically disordered proteins secondary structure propensities from alpha helix to beta sheet, but it's intrinsically and it's got a little bit of helix, and then the post-translational modifications make it into beta sheet. That's the closest example I know. It's a really beautiful paper. I suspect that the same thing is true for fold switching proteins, and who knows, maybe like in 2 or 5 or 10 years, we'll have real experimental examples.
Well,
Thanks,
thank you.
Lori.
All.
Wonderful. Thank you.
Thank you.
So happy holidays, and I hope you can
Yes,
join us
thanks.
in the first week of
Happy
January.
holidays. Happy New Year. Everything.
Thank
Right.
you.
Right?